Okay, so ladies and gentlemen, you are all welcome. I'm very sorry for the inconvenience. It's actually due to network problem. So patient, you are welcome. So what we are going to do now, we are going to solve some of the question paper. Really patient and Azoji, which kind of questions do you want me to answer? Please, I wanted you to go and write it on the WhatsApp group that this last link is the one that we are going to use for the revision so that at least more members will join the class. So for those that are here, okay, Kat, you are welcome. So now, what are we going to use? Are we going to solve first question paper? Are we going to do a general revision? If it is a first question paper, do you want objectives or we should do AC? So I'm listening. Which one? Okay, really, you want general revision. So, okay, AC, AC. Okay, sir, AC. Okay. So let's go for the AC. Let me check. I think I have the question paper. So let me try to check it here. Okay. Okay, so now I'm sharing the question paper, ladies and gentlemen. I'm happy the last question paper that we solved, some of the question paper also some of the yesterday's biochemistry questions came from what we have answered. So I think I will advise you after watching or after we are done with this lecture, I will also advise you to go back and watch the last video that I did for those that wrote their exam on that is yesterday. Yeah, so these are some of the questions that we are going to start with. Question 1A says, list three examples of each of the following. Roman figure 1 hexosis, Roman figure 2 disaccharides. Who can give me three examples of hexosis? What are the examples of hexosis? When we said hexosis, what does that mean? So please, I need answer for this. Hexosis. When we said hexosis, what does that mean? I'm listening. Oh, nobody knows the answer. Okay, you mean your question paper? So let me share it again. Okay, excellent. Fashion, you mentioned that hexosis we have glucose, fructose, and galactose. So generally, one of the major classification of carbohydrate, we classify carbohydrate based on their number of carbon. So we have trioses, that is those sugars that have only 3 carbon. An example of those sugars that have only three carbon, we have dihydroxyacetone phosphate and glyceraldehyde. And we have those sugars that have only four carbon, and we have those sugars that have five carbon, like pentose sugar, like in the case of ribose. Then we have those sugars that contain six carbon. So example that contain six carbon, we have glucose, we have fructose, and then we have galactose. So these are the examples of sugars that contain three carbon sugars. Then the next one is disaccharides. So who can give three examples of disaccharides? I'm waiting for the answer. Disaccharides. Three examples of disaccharides. Answer. Examples of three sugars that are found. Examples of sugars that are disaccharides. Three of them. Okay, John. Yes, we have maltose. Then one is sucrose, maltose, and lactose. Excellent question. So these are the three examples of sugar that is found. So then question 1b. Name one hexose found in plant gum. So can you give me examples of hexose sugar that is found in plant? I'm waiting for your response. So the answer to this is we have glucose. We also have galactose. So these are the examples of sugars that are found in plant gum that are hexose. That is, they have six carbon. Then question C. Disclose the lifespan of an animal cell. Disclose the lifespan of an animal cell. So we can discuss briefly what can you say about the lifespan of an animal cell. You see there is only 11 people that joined. Did you actually send them the link that this is the link that we are going to use? We only have 11 that joined. So we finished the exams very late, so some people might not be online. Okay, so no problem. Let's just try to answer the questions so that those that did not join will also join. So I said, the question says, disclose the lifespan of an animal cell. So, ladies and gentlemen, let me just say that naturally, the cells that are found in plant have their lifespan. Like for example, the red blood cell have a lifespan of 120 days. That means after 120 days, the red blood cells will be destroyed and then replaced with a new one. So generally, each and every cells in our body have their own specific lifespan. Why is it that the cells have their own specific lifespan? Is to protect them from the wearing and tearing of the cells itself. Because generally, cells undergo wearing and tearing. So that is why each and every cells in our body, although not all, we have some specific types of cells that are not divided and replaced with new ones. Like for example, the heart cells, that is the cardiocytes, and the brain cells. They are the cells that cannot divide; they only enlarge in size. So those cells that doesn't have lifespan. Their lifespan is when we die. So when we die, or when our brain cells or the heart cells are getting problem, so there is no mechanisms that will actually cause the death of cells and then replacing them with new one. So what I wanted to us to understand in this case, some apart from the cardiocytes and the neurocytes, all other cells have their lifespan. That is, they have a specific time that they will be destroyed and then synthesize the new ones and replace them. So that is just briefly on that. So I think this question one is very, very simple, very simple. Then question 2a. With the aid of a structure, describe the chemical nature of an amino acid. So who can tell me what are the major groups that are found in amino acids? I'm waiting for your response. Yes, good evening, Diago. You are welcome. I didn't see you today, so you didn't join early. So I'm asking, what are the major groups that are found in an amino acids? I'm listening. Please write them down. Answer it. Okay, you're not seeing. So let me share the screen again. It's like some of you are not actually seeing my screen. So I'm sharing it now. The head of the structure describe the chemical nature of an amino acid. So what are the four groups that are found in an amino acid? I'm waiting for your response. I am waiting for your response. What are the four major groups that are found in an amino acid? If you are asked to describe the amino acid, what are the four major groups that attach to the central carbon? No, Kaulat Ahmad, we already answered. There are four major groups that attach to it, and that is why that carbon is called chiral carbon or stereogenic carbon, because it has four different groups that attach to it: hydrogen, amino group, carboxylic group, and free hydrogen. So because of this chirality nature of the central carbon, that is why they undergo isomerism. And then the central carbon, as I said, is called chiral carbon. So all those amino acids have that chiral carbon except glycine, because in the glycine, there are two free hydrogen. But a carbon is qualified to be called chiral or stereogenic carbon when it has four different groups attached to it. So that is 2A. Then, so this question 2A, is very simple, and look at it, is 5.5 marks. The question is just asking you to draw the structure of the amino acid, the general structure of an amino acid. You write the central carbon, carboxylic group, amino group, R side change, and then free hydrogen. John, you are correct. Then question 2B. I want someone to answer this question on the comment section. Number 2A, sorry, 2B. Enumerate 4 techniques used in protein purification or in the purification of protein. I'm waiting for your response, please. Do you mean nobody can answer this question? So let me answer the question. Number 1, we have ammonium precipitation. We have affinity chromatography. We have column chromatography. We have SDS phase, that is sodium didecyl. And then uh, we have high performance liquid chromatography. We have electrophoresis. All these are the techniques that are used of proteins. So I hope this is. Please, I'm not seeing your comment on the comment sections. Are you with me? Are you with me? I said, are you with me? We have amino acid sequencing method. That is Edmund sequencing method. So all these are the examples or are the 4 techniques that are used for purification of protein. So now the next questions that we are going to answer is on the question says, question C, mention 3 proteoglycans found in connective tissue of humans. Please, I don't know. Are you with me? Because I realize that you are also having a network issue. Are you with me? I want to see your response. Please, are you with me? The network is not stable. It's like the network. So what are we going to do? Actually, I am also having the network issue here. Can we continue the class, or maybe we should just leave it? So what are you going? No, actually, it's not about the issue of Google Meet. The problem is that my network problem. I tried my Airtel MTN and they are giving me headache seriously. Is the network stable from your side? So is there still network? Is the network okay? Seriously, we are having network issue.